If you think about a new technology, you have to develop an understanding of it, and your most critical asset at this point is your data. And to apply that data and to really understand that data, we need meetings like this. And our goal is that all users and potential users stay connected to this, because that's how we'll move forward. And it isn't just what's the right patient, it's what's the right technique and what are the right centers. And so this is part of that process. So in the spirit of that, today we'd like to present our recently completed FDA pivotal trial. It is a trial that has been submitted to the FDA. It will be, it's under review right now. It has not been published, so you're going to be seeing this data really for the first time. Give you a little bit of background here on our clinical experience. Torax Medical started in about 2003. Spent the first 4 years just developing the LINX device, prototypes, doing benchtop testing, doing animal testing, studying the literature, trying to understand the things that Dr. Meester was just talking about. And when we got into the literature, it was interesting. The papers that made the most sense to us all had the same name, the T.R. de Meester. And we said, who is this guy? And we went out and met him, because we were thinking about this in a mechanical way without the clinical perspective, and we were really able to partner with his approach from a clinical perspective, apply it to the engineering, and over the last few years really developed this technology. So our first implant was in January 2007, so over 4 years ago. Torax Medical is based in Minnesota, so the first human implant was done in Minneapolis over 4 years ago. Since then, we've had over 200 implants in over 5 countries and 20 centers. So this experience that we need to control is getting bigger, but we still need to stay close. It's still too early to let this go unsupervised. We've had 3 papers. Our first paper was published in 2008 in gastrointestinal endoscopy, and it was really all of our preclinical work. So it got into how did we come up with these forces? What are the yield pressures? And what is the histopathology of this device when implanted in animals? So all this was published. The second trial was our initial clinical experience. I don't remember how many patients, but it was our first clinical experience. The last trial, Annals of Surgery, really looked at the 2-year experience. So really, what is this device and what is it capable of? And I don't know if Luigi, he's here. So Professor Bonavina is here, who really led this paper, has the biggest experience, and really gave us the confidence to do our pivotal trial, which is the U.S. trial, even though this study also had U.S. centers. So the next steps for us is the European registry. And we need to continue to build this clinical story and have the same opportunity to look at these experiences as we go, so we don't start to put this in all these different centers and start to have problems. And we say, well, what patient did it go into? What center did it? What was the procedure? We don't want that. That's where this registry is very important. It's going to help you all stay connected to this and have the information to build this procedure. So the LINX technology, Dr. Meester talked about the therapy gap, and that's really what this device is intended for, is this gap patient. How can you augment someone who has a deficient sphincter, but do it in a way that's simple and reproducible? Can you engineer out some of the variabilities with current anti-reflux surgery? That was the premise. The idea here was we weren't trying to squeeze the sphincter. And so if you look at manometry after LINX, you won't see necessarily an increased resting tone. We're trying to resist that effacement. When there's gastric wall tension due to a gastric challenge, it wants to efface that sphincter. The device, it has to overcome that magnetic bond. And the theory is it can't. It doesn't mean you will not be able to have other physiologic functions like vomit and belch. And these are things that we wanted to really look at in this trial. Can you resist the sphincter opening? Does that mean you have less reflux? And if you do that, are the side effects created or are they not? So the overall therapy goal is, can we take a patient earlier in their disease stage when they're less tolerant of side effects, augment their sphincter, relieve the symptoms that they're not getting on PPI therapy, but not create new problems. So this is the construction, and if you think about augmenting something, you need a force. So how do you do that? Is it going to be battery powered? Is it going to be elastic? What's really unique about magnets is they are their own energy source. And we use a special magnet, neodymium iron boride. It's a magnet that won't wear out. And we can control the force. So this is the magnet, this gray area, which is a core of each of these beads. We can control the force by the mass of the magnet. So all the 4 years of animal work was just titrating that force to make sure we had the right amount to prevent yield, gastric yield, but not so much that it affected peristaltic function, normal swallow function. So it's really simple. It's a series of magnetic beads that are linked together. If you don't have these little arms here, magnets, you put them on the table, they're all in a pile. They don't behave. So what these arms do is it makes them behave in an annular fashion. And when you create, when they're all touching, which is their natural state, there's a bond between each of these that the gastric pressure has to overcome. And in theory, it can't. When you swallow, higher pressures are created, the magnetic bond breaks, and the physics of magnets is once there's any separation of the magnets, the force exponentially drops. So this configuration here, you have little force, and you're able to swallow this, and then it returns to this configuration. Now the force isn't zero here, it's still attracted to the adjacent bead, and that's what brings the device back together after you swallow. So these are all small things, but were thought out over years of work in bench testing and animal testing. So what we'll show you here is one of these animal tests where this is just a porcine stomach. This is pressure. Obviously, and you just see that if you pressurize it, you see the fundus and the facement starting right here, and it starts to leak. Okay? If you place the LINX device around, watch the pressure. So that's really it. That's all we're doing. That's the simple mechanism by which Dr. Meester was describing this morning, this effacement. So the way that looks, you know, anatomically, if you look at the, so the device is placed at the GE junction, and we'll talk about some of the surgical technique here later today. And when you see it here, it's very important. We're not trying to squeeze that esophagus. We don't want to impart a chronic force. We're simply trying to resist it from opening, if that makes sense. When you swallow, we talked about this, the magnetic bonds are broke because of the higher peristaltic pressure and the food is allowed to enter the stomach. And there's enough residual traction that the beads reapproximate after the peristaltic pressure is gone. So if you look at this in an actual human implant, and you know we have what I'll show you is a barium swallow here, which just gives you another look at what that looks like. So first the liquid bolus here. You see one bead open here. You see, obviously, a food bowl is coming through, and you see the beads be pushed apart. And once that food enters the stomach, they reapproximate. So if you take an X-ray on a patient that doesn't have a, just a plain chest X-ray, you'll see the device, and you see all the beads touching in its at-rest state.